Thank you very much for the organizer for inviting me for the conference. Since we are already after lunch, so I will have a short presentation. I'm from Stem Cell Bioengineering Unit, Mostokowski Medical Research Center in Warsaw, in Poland. And the work was financed by the Wrocławian Research Center AIT+. And I will talk about reciprocal interactions between iPS-derived neural cell-seeded modified collagen scaffold and nerve tissue. The aim of our study was to build up such kind of platform suitable for in vitro investigation of the mutual relationship between endogenous nerve tissue and artificial scaffold-based 3D neural stem cell culture. The tools that I was using was the iPS cells differentiate later on, the collagen scaffolds, the neural tissue was the DRG and the spinal cord organotypic slices. I have co-cultured such a system and investigated. And the project is still already running, so I do not have the final results. We have heard today a lot about the neural regeneration, and I would like to just to add that we can apply cell scaffold hybrid system for neural regeneration for example, replacing damaged injured tissue or supporting regeneration in vivo by applying the scaffold seeded with stem cells, either, for example, in the injured or damaged spinal cord or injured brains. We can create the tissue models in vitro by creating the biomimetic niche for testing disease and developmental processes. And in these 3D models, we have to have the scaffold, we have to have some extracellular matrix proteins, we have the stem cells, sometimes the neighboring cells, because the cell contacts are very important to behave for the proper behavior of stem cells. And such a cell scaffold hybrid system can be as well applied in toxicity and pharmacological testing. To develop such a system, three-dimensional biomimetic niche, we have to have cells. We can have the cells from embryonic or iPS cells. We have to differentiate them, seed on the different kinds of scaffolds, hydrogels, nanofibers, collagen scaffolds, and then co-culture with the nerve tissue. As we all heard today and yesterday, the discovery of reprogramming was very successful for the application in such a cells in clinical application. So we can have the patient, we can isolate the cells for reprogram cells, and later on differentiate for different types of cells, and then use the cells for the therapies or disease models or drug screening. And the methods for iPS cell obtaining are more efficient or safe, and there is no good solution for. We have the very efficient method based on virus method, but they are not suitable for clinical application. From the other side, we have the episomal vector, small molecule or protein that could be applied in a clinical, but the efficiency is very, very low. And iPS cells in neurological disease can be used for better understanding of disease biology and potential pathways for investigation. We can develop possible diagnostic tools. We can select and select compounds to be alleviate the disease phenotype in vitro, or we can translate into clinically relevant therapies. And the biomaterials added to the system can have the broader point of view for such a system. I was working on human-induced pluripotent stem cells. It was the stem cell line episomally derived without viral and feeder system. So it was feeder-free system, serum-free system as well. So it is nice to apply this in a later clinical application. The cell line was derived from cord blood-derived CD34 plus progenitor cells, and in culture it expressed OCT4, TRI-160 and SSEA34 proteins, as well as NanoXOX. And after differentiation, I was able to obtain the cells that are from three lineage, neural lineage. I could have the glial progenitors, the GALC means the oligodendrocytes in the culture. I could have the GFAP or beta-tubulin positive cells. The cells very nicely differentiate into neural cells, and a lot of cells in this culture stadium have the nestin markers. And after prolonged differentiation, these cells could very nicely differentiate into neuronal phenotypes. The double cortin positive cells and beta-tubulin 3, the NF200 and synapsin TH positive cells, so and the GFAP of course cells we could obtain in the culture. And the next step of our work was to obtain the scaffolds. We have the porcine collagen type sponge shaped scaffolds made by freezing method, and after creation of the scaffold, the scaffold were functionalized, modified by carbodimide and NHS to improve mechanical and thermal stability. And prior to cell culture, the samples were sterilized and functionalized with laminin. And physicochemical properties of collagen scaffold. The non-cross-linked scaffolds were very easy degradable by collagenase, while the cross-linked scaffold were resistant for the degradation and have very nice surface area of the pore and pore volume, what give good adhesion and allow substantial surface interactions. The collagen non-cross-linked scaffolds, unmodified scaffolds, were collapsing in the culture, and the culturing of such a scaffold was not able. And here, just to show you the spectrum of collagen scaffolds, unmodified and modified, that it shows that the modification prevent or made better the helical structure of the collagen itself, and the degradation of the scaffold immersed in aqueous solution was quite good, and it also shows that the scaffolds can be biodegradable. And we have as well the three types of scaffolds. They were different by percentage of collagen used for the production and the temperature of freezing, and we choose the one type 1 scaffold. And after seeing our cells on such a collagen scaffold, I saw that the cells adhere to the scaffold very nicely. The scaffold have very nice imaging properties. So the autofluorescence of the scaffold is not very huge. So with the confocal microscopy, it's really easy to visualize the cells. And the cells, after sitting on the scaffold, still have the markers that were in a culture. but sometimes the cells create clumps, so the adhesion was not the best. So we wanted to functionalize with a laminin, and after deposition of laminin on the scaffold, we could have the cells that create a lot of protrusions, and the spreading of the cells on the scaffold was really better. And as well, here you can see the neuronal cells seated on the collagen scaffold that have the double cortin and map to markers. And later on, we have co-culture, such a hybrid with with the organotypic dorsal root ganglia and spinal cord slices. And first of all, we cultured scaffold itself without cells with a DRG, and we've seen quite nice, very long protrusions from the slice, from the DRG to the scaffolds. The axonal outgrowth was very nicely visible, and in very long distance. However, the culture of such a system was very, very long. It was like 4 weeks in a culture. But we could see the very nicely migration of S100 beta cells into the scaffolds, and sorry, and the axonal outgrowth from the DRG into the scaffold. Here once again, the extension of the axonal outgrowth to the scaffold, and the migration of the Schwann cells on the fibers NF200 beta-2 so on the Neuritt fibers. And we have checked whether the laminin would promote the protrusion, making the protrusion of the cells. So we tested the control conditions. So these are the dissociated DRG and neurons on the control cover slip covered with laminin, and we counted the cells with a long, short or non protrusions on such a system, and we have checked three kind of neurons co-cultured with scaffolds, and we have the result was that the laminin enhanced the number of neurons having the long protrusions, while on the laminin, on the scaffolds without laminin, we have the highest number of such a round cells without protrusions. And the model of the organotypic spinal cord slice culture was developed by, was used by our collaborators, Professor Gary Brook from Ahana University, and they, first of all, they checked the different kind of scaffold, the fibrin gel or collagen scaffold with the pores, co-cultured with the spinal cord organotypic slice culture, and they show that the migration from the spinal cord through the scaffold is really good, and the axonal outgrowth is very good, and the Schwann cell migration is as well positive, while on this fibrin gel they couldn't observe the GFAP migration, but very nicely visible the migration of the macrophage microglia cells. So when we were seeding our cells on the scaffold and then co-culture such a hybrid with a organotypic slice or spinal cord, we could see that the cells do not, unfortunately or fortunately, do not migrate into the slice culture. We labeled the cells by human mitochondria or CMFDA tracer, green tracer. This staining was not the best because the CMFDA is releasing from the cells and gives a huge background into the scaffold. But the human mitochondria was chosen as a choice for the next results, next experiments. And yes, and here we can see that the scaffold is fully packed with iPS-derived neural stem cells, but the migration into the slice culture is almost none. And here we have the all scaffold. Here is the slice culture, and we have found that the cells are evenly distributed throughout the scaffold, but they are aggregating in the long distance away from the slice culture, and there is no migration from the scaffold into the slice culture. However, there is the migration from the slice culture into the scaffold. Here we can see that the beta tubulin 3 positive axonal outgrowth is visible from the organotypic slice culture into the scaffold, and the GFAP positive astrocytes are as well migrating from the scaffolds from the slice into the scaffold as well, and ED1 positive cells as well. So in conclusion, I can say that our platform that we told it is some kind of platform is suitable for testing the interrelation between the transplanted cells and organotypic tissue explants. We have observed cell migration, but it was the unidirectional from the neural tissue into the scaffold, and we observed the axonal outgrowth as well in unidirectional manner from the slice culture into the scaffold, and the functionalization with the laminin enhanced the axonal outgrowth from the in a DRG organotypic culture, and whether the presence of human iPS cells attract the mobility of the migration from the slice into the culture needs to be still verified. Thank you. I would like to acknowledge Dr. Krystyna Pietrucha from Łódź University of Technology. She made the scaffolds, and Professor Gary Brook from Aachen University was responsible for organotypic slice culture, and our team in Stem Cell Bioengineering Unit at Mosakowski Medical Research Center in Warsaw. Thank you very much.